Hello, everyone. I'm Priya from Optics11 Life. I welcome you all for our webinar today, and I'm really excited to have you all. More than that, I have a very special guest today with us, Dr. Remi Peyronnet. There he is. Thank you so much for joining us, Remi. He's head of cell biophysics, working in Freiburg, and he has a massive experience in mechanotransduction and mechanosensors. I can't wait to hear his research today. Thank you so much, Remi. Floor is yours.
Thank you very much for this kind introduction. I'm very happy to be here today and to have the opportunity to share some of our results. Let me share the presentation. Here it is. And yep. So yeah. We are today going to see a few experiments that we did on human atrial fibrillation stiffness and mechanosensing. I hope you hear me well. If you have questions, please do not hesitate to interrupt me, and I will go through the slides here, starting with our university. We work in Freiburg, Germany. This is here south of Germany. We have a nice place to work in, and we have a number of projects focused on rehydration, focused on food diets and exercising. We have a very strong focus in cardiac sciences. And a lot of work has been done to better understand the heart so far. And most of it is based on the understanding of what the electrophysiology does to the mechanics. So essentially how an action potential can drive a contraction. What we do is more focused on the feedback, like the fact that mechanics can alter cardiac physiology, electrophysiology and physiology. So this is going to be my focus for today. I will briefly introduce the different projects that we are driving in the group. And in case of this could be of interest for anyone, I'll be happy to tell more later after the talk, in case of you're interested. So we drive a number of projects that are mainly around mechanobiology and better understanding of mechanosensors or load-bearing structures and what this could do to the cells and the tissue. We have projects focused on subcellular mechanobiology and one on nuclei. Here we worked in collaboration with the Max Planck Institute to better understand what essentially epigenetic modulations can do to lamins in the nucleus. So if the acetylation of lamins is changing mechanical properties of the nucleus and shape, here you can see that when you stop acetylating lamins, there are fragments of the nucleus that are forming and some nuclear material that is escaping in these small vesicles. In fact, there are proper holes inside the nucleus that are forming with these vesicles going out, and where you can have some genetic material escaping from these vesicles when there's a lack of acetylation. We were wondering if this could alter the mechanics of the nucleus, and using the nano-indenter, here we showed on freshly isolated nuclei that indeed when there is a default in acetylation, the structure of the nucleus and the lamins are softer. Nuclei are softer, and we think this contributes to explain that there is here formation of these vesicles. We are also interested in aneurysm. We don't focus only on the heart, but also on vasculature. What are aneurysms? They are local deformation of vessels. And this local deformation of vessels, they are essentially due to local change in mechanical properties, regional stiffening, regional softening of the tissue, which is a big problem. If these large vessels rupture, obviously this leads to major bleeding. So we try to better understand these mechanical properties regionally and at the tissue level, at the cell level, and we try to understand what role mechanosensors at the cell surface can do in this tissue modeling. Last but not least, we work on embryonic heart and try to better understand the embryonic heart functions. This is a little bit on the side, but I wanted to highlight it here because this is a pretty, we think, interesting use of the nano-indenter. So this is an embryonic heart, mouse embryonic heart, with the atria, with the aorta, and the ventricles. This is about a few millimeters in size. I'm sorry, I didn't put the scale bar here. And it beats nicely after excision. So you can appreciate motion of the ventricles and of the atria. Question is, if here you want to assess rhythm, if you want to assess force of, get an idea of the force of contraction, this is pretty difficult because here we talk about millimeter size, for whole hearts, we can use balloons inserted in ventricles to assess regularity of the beating, force of the contractions, and so on. All these kind of things are not possible in the embryo. So the idea was to put on top of these embryonic hearts, like the cantilever of the nano-indenter, and to press a little harder, step by step, on the tissue, on the whole organ, actually. What do we see? We see here that we can get contractions. We can get a pretty good readout of the contractions. So if we look carefully at one of these spikes, you can really see a nice signal with a wonderful signal-to-noise ratio. And from this, we can extract maximum speed of contraction, of relaxation, time to peak, plenty of parameters, peak amplitude, obviously, which give an idea of the work of this this little organ. And if we analyze step by step, here looking at steps individually, you can very easily see that the amplitude here are, the amplitudes of contraction are different. So the more we indent, the larger the amplitude. So this is very interesting because this is really well known at the whole heart level, at the single cell level. The more you stretch the material, the cardiac tissue, the stronger it will contract. That's exactly what we see here, which is, we think, a good control for the quality of the readout. Obviously, we record the force perpendicularly to the tissue. So this is not as relevant as measuring ejection fraction and these kind of parameters, but it helps us to see if the heart beats regularly and to monitor the amplitude of contractions. So this is really in a nutshell the different projects that we run in the group. Today, I'd like to focus on stretch and cardiac arrhythmias. We're interested in one specific cardiac arrhythmia, which is atrial fibrillation, AF. What is atrial fibrillation? Atrial fibrillation is a complex disease with the influence of a number of environmental factors. The exact cause of AF are not known, but what we know is that there is an electrical impairment. Action potential shape, for example, is changed. There is also a contractile impairment. The atria are not beating properly. They are mechanically overloaded. They are often dilated, and this is one of the features here that is going to be of interest to us. There is also presence of a structural remodeling, fibrosis, and this is also something that we are very interested in. Fibrosis is the overaccumulation of extracellular matrix. This is stained in red. You see that in patients here having atrial fibrillation, there is a lot of this fibrotic tissue compared to the control tissue. What does it mean? It means that the tissue is stiffer here. It will deform differently. Forces will propagate differently, and this will lead to stretch. The tissue is likely to stretch around these very stiff areas compared to the soft ones. And we hypothesize that the stretch can alter the electrophysiology of fibroblasts, and this will alter fibroblast function. If we are right, these cells, these atrial fibroblasts, they should have mechanosome cells. There are different mechanosome cells that are known, structural ones, like the cytoskeleton, linkage proteins like integrins connecting the extracellular matrix with the cytoskeleton, and mechanosensitive channels. There, I'd like to focus on these mechanosensitive channels. What are they? Very simply, if you apply membrane stretch, in-plane stretch will increase the open probability of these channels. Same if you increase the membrane curvature, you will open these proteins. We use primary cultures and a cell line of human atrial fibroblasts from the right atrium. And because these are channels, we use the patch clamp technique to record them. We use pressure that we apply through the patch pipette here that is attached to the plasma membrane of these cells. And we use this negative pressure to change membrane curvature and activate these channels. So what do we see? We see indeed that when we stretch here, we generate a current. The more we stretch from 0 here to minus 80, the more current we see. So we can, by using a knockdown experiment, we can see that this current here that is in blue in control condition from 0 pressure to minus 80 millimeter of pressure with the average current shown here, pretty nice current in blue, is almost completely abolished when we silence piezo1, which is a well-known stretch activated channel. So most of the activity that we see here is generated by this channel piezo1. There is another channel here, which is a potassium selective channel, which is called BK. And I will not enter in the details, but what we found is that these two channels are functionally coupled. The calcium going inside the cell via this piezo channel will activate this potassium channel that is calcium sensitive. I'll be very brief on this aspect. This is not the topic for today. We know that these two proteins do not colocalize, so there is a functional coupling with no physical interactions. That's one aspect of what this piezo channel can do, activate this potassium channel. But there is another function. And what is important to highlight is in the disease, here atrial fibrillation, we have this piezo channel that is more expressed compared to fibroblasts from patients without this arrhythmia. And interestingly, on the functional aspect, when we again look at the pressure from 0 to minus 80, and as a readout, we use the average current, we see that there is a lot more current in cells from diseased patients compared to control patients. So more proteins, apparently more activity and more mRNA in the diseased cells. That's interesting, and that's something we wanted to understand better. So as a recap, we have indeed stretch and two mechanosensors here that are talking to each other, regulated by dysregulated by the disease. And we want to understand better the function, especially the function of piezo and related to tissue remodeling. What do we see? What is the second function of this channel piezo that we spotted? What we saw is that there is a remodeling of the cytoskeleton of effectin. So here in this little graph, we see the phyloid intensity of control cells, of cells with piezo being overexpressed, and of cells having piezo knockdown. So almost no more piezo expressed in these cells. And what we see is the more piezo expressed, the more phyloid signal we see, the more effectin. And when we knock down piezo, there is less effectin signal detected. That's one thing. The other thing is that there is a reorganization of the cytoskeleton. Essentially, when we overexpress piezo, effectin bundles tend to be better ordered, more organized compared to the control condition. Also, actin bundles are thicker. So if there is such a remodeling of the actin cytoskeleton, if this is true, we should be able to spot a change in cellular mechanical properties. If the load-bearing structure that is actin is so much remodeled, we should spot some changes in the cell stiffness. And that's what we wanted to test. We used the nano-indenter. I'm not going to introduce further the principle here, And what we do here is to record here the indentation, the force that is generated here following the indentation. So we get the Young's modulus, and I'm going to refer to the Young's modulus as stiffness. So what do we see? When we overexpress piezo, we see that the cells are indeed stiffer. So it's not only about like imaging the cytoskeleton and seeing that the cytoskeleton is remodeled. This has a functional impact on the mechanical properties of the cells. Cells are stiffer, and if we knock down piezo, cells get softer. So we have a strong remodeling of cell mechanical properties here, fibroblast, cardiac fibroblast mechanical properties. Great. So we wanted to go a little deeper and see if this would apply to other channels that are similar to piezo. So we used an expression model like hex cells. We overexpress piezo, and again, we see that when we overexpress piezo, cells get stiffer, as we saw in the atrial fibroblast. And now what we did was to overexpress a channel similar to piezo, TRPC6 here, which is also a channel which has a similar selectivity. It's cation non-selective. It's also involved in mechanotransduction. So some very similar biophysical properties and also involved in similar pathways. And here, when we overexpress it, we don't see changes in cell mechanical properties, cell stiffness. So it looks like that at least this remodeling of the cytoskeleton is not something that is seen for all kind of channels. It looks that is something that is more specific to piezo. Then, as I said, piezo is a channel. So we wanted to understand if this cell stiffening would be due to the channel conductance. So to test this, we use an antibiotic, which is very well known to block all kind of stretch activity channels that are cation non-selective. This is streptomycin. And when we have streptomycin, when we block the channels at the plasma membrane, we see that the effect, the cell stiffening, is still there. When we overexpress the channel in red, we get the increase in cell stiffness, as we see when the channel is not blocked. Second, as another kind of control, we wanted to see if, in case of we miss something, in case of there are channels expressed on endomembranes, would a change in the cytosolic calcium would change the cell stiffness. And a very well-known target of calcium is calpain, which is calcium sensitive. It's a protein which is calcium sensitive. And here, if we block calpain, we still have the piezo-induced increase in cell stiffness. So this is not calcium, not calpain mediated. So what could it be? Piezo has been related to integrins. So we wanted to dig deeper in this direction and see if integrins could be involved in this piezo-induced cell stiffening. So we either activated integrin beta 1 or inhibited integrin beta 1, being known to be related to piezo. And when we either block or activate the integrin, we don't see any difference in our readout. When we overexpress piezo, we still see the stiffening. I'm sorry, I made a mistake here. When we activate the beta integrin, we get a stiffening of the cell, which is expected, and there is no more difference with piezo. So this is our positive control. When integrin beta 1 is activated, there is an increased stiffness, which is more or less matching, which is not significantly different from the cells that are overexpressing piezo. When integrin beta 1 is inhibited, we still have the piezo-induced stiffness. So meaning that piezo-mediated stiffening of the cell doesn't require the activation of integrin. We went further and we investigated the downstream signaling of the integrin signaling pathway, and we targeted the focal adhesion kinase. We inhibited FAC, and when we inhibit FAC, piezo-induced increase in stiffness doesn't occur anymore. So if FAC is inhibited, piezo is not capable of increasing cell stiffness anymore. We tried the raw associated kinase, and this didn't change the effect of piezo. So piezo is still able to make the cells stiffer. So this is true in HEC, and this is what we see in our human atrial fibroblast. As a little recap, piezo can modulate cell stiffness. This requires focal adhesion kinase, not piezo conductance at the plasma membrane, not the integrin beta-1 activation, not the calpain, not ROCK. So we went on with another kind of observation. Everything that I've shown so far is based on cells that are overexpressing piezo. We have a plasmid which allows us to overexpress piezo and overexpress GFP. The two proteins are not fused, but they are expressed together in the cells. So if the cell is green, it means that the cell has piezo. So that's what we saw so far. But interestingly, the non-fluorescent cells in the same dish have also a piezo-induced increased stiffness. So what does it mean? It means that when we have one dish, we don't transfect all the cells. Actually, our transfection rate is quite poor because of these primary cultures of these atrial fibroblasts that are very difficult to transfect. So when piezo is overexpressed in these cells are stiffer. But we see that the neighboring cells, not overexpressing piezo, they get also stiffer. That's what we see here. How is that possible? So we suspected a paracrine mediator here. And by reading the literature, we found a candidate highlighted by the group of Neil Turner, which is IL-6, which is an interleukin, which is involved in inflammation. And here the expression of IL-6 is correlated with the expression of piezo. So we measured the amount of the concentration of IL-6 in our culture medium in controlled cells, not overexpressing piezo, in the cells overexpressing piezo. And to our surprise, although our transfection rate is really low, we saw we saw a significant increase in IL-6. And we see that if we block IL-6, we don't have any more piezo-induced increase in cell stiffness. So it seems that in these non-transfected cells, we get the signal transmitted via interleukin 6. One last observation that I want to highlight today is that piezo doesn't only change cell stiffness, but also alters another cellular function. And here, what is well known is if you seed cells on soft or on stiff matrices, you will change cell stiffness. And so we were wondering how these cells would adapt to matrices of different stiffness. And here we used hydrogels, stiff in dark blue and soft in light blue. And as expected, when we seed our human atrial fibroblasts on stiff matrices, they get stiffer. If we seed them on soft matrices, they get softer. So that's exactly what is described in the literature. Cells can adapt their cytoskeleton to their mechanical environment. Now, what happens if we knock down piezo? If we knock down piezo, this adaptation to the stiff matrices doesn't happen anymore. Here you can see that there is no more significant difference. These cells seated on stiffer matrices without piezo are not able to adapt, to have their cytoskeleton adapting to their stiff environment. So piezo contributes to stiffness sensing, to matrix stiffness sensing in human atrial fibroblasts. And this is pretty interesting because substrate stiffness is known to alter a number of cell types. So fibroblasts are known to respond to matrix stiffness, but also macrophages, neonatal cardiomyocytes, induced pluripotent stem cells, derived cardiomyocytes, adipocytes, neuron, and also cell types. Like cell functions are strongly modulated by substrate stiffness. There are functions that are not cell type specific and still modulated by substrate stiffness, the direction of cell migration, cell division, cell differentiation. And if we focus more on fibroblasts, cytoskeletal remodeling, as I showed, secretome, the way one of the important functions of fibroblasts is to secrete ECM proteins, and also the phenoconversion. And when the tissue is diseased or injured, this fibroblast will convert into phenoconvert from fibroblast to myofibroblast. And this can lead to fibrosis, and that's why we are very interested in this aspect. So if I summarize, we have fibroblasts here from healthy tissue, and they can differentiate in myofibroblast, essentially characteristic of diseased tissue. This can be triggered by mechanical stimuli or by biochemical stimuli. And here we see that piezo is present. Piezo conductance doesn't lead to the myofibroblast differentiation, but here can trigger the production of IL-6, which in turn can generate stiffer cells. And this is exactly what we are after at the moment, is this kind of channel, stretch activity channels involved in myofibroblast differentiation. I want to thank the people from my group, especially Dorothee Jacob and Alexander Claesen. They worked on the channels, the channel aspect, the electrophysiology, and the people who heavily worked on the mechanical property characterization are Ramona Emig here, who did most of the work with Wiebke, Knott, and Mario Krusik also contributed to this work. I also want to work our director, Professor Kohl, Professor Ursula Ravens, who is supporting a lot of our projects, and all the surgeons and patients who make this possible, because this is mainly based on human samples. Thank you for your attention.
Thank you so much, Remi. It was really interesting work. I do want to ask you a couple things, as I'm very intrigued, and I hope I would still have my own presentation afterwards, because it's perfectly in alignment with what you shared on your last slide. Firstly, so just so everyone who is attending the webinar knows, which one of the nano indenters are you actually using?
Yeah, I should have mentioned this. This is the Tiaro. This is the Tiaro.
So is it like integrated with one of your in-house microscope?
Yes. So we have this on one of our microscopes with anti-vibration systems, but actually we move it quite a bit. So most of the time we have the Tiaro with a simple microscope with fluorescence. But when we need to go a little bit more fancy, we couple this with also device to stretch the cells or a confocal microscope.
Oh, nice. I didn't know that. Well, that shows the usability, quite adaptable to the different microscopes as well. Though, while looking at your work, I mean, it's quite intriguing. I'm also curious, what would be the next step now? Like, as you have mentioned, that mechanical stimuli or the mechanical properties does play a very important role. So how do you think that this would evolve in the coming project or the next step in the research that you are heading towards?
So like what I showed mainly is cell work, and we try to bring this now to tissue level. So we suspect that these proteins are playing a role in tissue remodeling. And fibrosis is a big burden. Like when your heart is fibrotic because, for example, it's infarcted because maybe of the intervention of a surgeon when you have atrial fibrillation to treat at the end atrial fibrillation, to stop atrial fibrillation, surgeons, like, they burn the atrial tissue, they make a scar in the heart. And the properties of this car is really important for the outcome of the surgery. So we try to better understand this. So now we try to bring this research to tissue level. So we go and grow tissue in culture and see if we can modulate the fibrosis. So here we want to combine the local mechanical property testing that we do with with the Tiaro. We want to combine this with a tensile tester to get bulk tissue mechanical properties. So get a readout at the cell level, but get a readout at the tissue level as well.
Nice, quite interesting. I look forward to your next publications. And once again, thank you so much for joining us. I would keep the platform open. Please, everyone, whoever has questions, don't be a shy audience. Feel free to use the Q&A button at the bottom of the screen and ask any question you would like. To move forward, I would proceed with answering maybe some of the curiosity. What is this Tiaro nano-indenter? How do you combine? Or what other nano-indenters could be possible that you could use? We proceed with the next presentation now. So, like we already discussed that we would be talking today about mechanics matter, and Remi has done a great job. I probably don't even need to share anything, but with the follow-up concern, how you can use this nano-indenter for yourself, I would like to proceed with one of the quotes that it really struck me, like it really held me there in a moment, and I was like, wow. And it goes like this: mechanobiology is the elephant in the room. It has long been overlooked, despite the fact that biomechanics is known to influence many functional features of biological life. I would say last part again, influence many functional features of biological life. It really aligns with what Remi has shared just before. And when I think of biological life, I think of one thing: cell. A stem cell can actually divide into muscle cell, nervous cell, blood cell, bone cell, epithelial cell, then the list goes on. And you know, the decision is made by one simple thing. That is like biomechanical cues that are basically playing an orchestra and deciding the cell's fate. So given that, it is very important that we understand how that cell mechanobiology, or what a plain key role this mechanical cues are playing in this. Not only in cell mechanobiology, like even we saw that Remi wants to proceed now with the tissues, and knowing the information, taking that information from cell mechanobiology, and now he's taking it to next level. Similarly, when we go to 2D or 3D mechanobiology, whether it's hydrogels, tissue scaffolds, porous scaffolds, printed scaffolds, name any scaffold. Given the field that we would be targeting, any application, whether it's skin and wound healing, whether it's cartilage, whether it's muscle reconstruction, drug delivery, and so much in the regenerative medicine. In the end, it is the mechanical property of the hydrogel tissue scaffolds that is going to play the role. That is going to decide the success of the result we get inside the lab. An in vitro experiment has to go to in vivo and eventually to be tested as a scaffold, right? So how everything is coming together is basically mechanical properties. Does it surprise you? Probably not. I mean, you're going to go to the supermarket and buy an avocado today, and the first thing you check is the mechanical property. How stiff is it? Am I going to eat it tonight or a week later, right? So what is the need of art? I would like you to ask one question. Do you work with cells, hydrogels, extracellular matrix, tissues, organoids, spheroids, or any engineered tissues? Because if the answer is yes, my next question to you would be, do you do mechanical testing? And if not, why? Think about it. Why are you not doing it? Is it too complicated to go on a nanoscale, or is it too soft to go for a bulk testing? Because I'm going to help you find that piece. Let's find that missing piece today. How are we going to do it? Look at the Optics11 Life Technology. We use a very simple principle. In this graphic, you see a probe, and there is a free diving board kind of structure that is a cantilever at the end of it with a spherical tip. There is a sample right in the bottom. The probe will come down and come in contact with the sample, basically like testing that avocado. And similarly, with this information of cantilever bending, we can understand how stiff or how soft or how is the mechanical property of your sample. The simple information that would define or basically differentiate between a healthy and unhealthy tissue and so much more. What you can do with Optics11 Life Technology is using that simple principle, you can use the system in 3 different modes. Displacement control, load control for creep measurements, depth control. Hydrogels are known to be depth dependent. They might be stiffer outside, but softer inside, or vice versa. Not only that, the cells, nuclei has a different property than a cytoplasmic fibers, right? So given all these controls that are provided, you can even see your profile in a static or the dynamic oscillatory profile. Given that you get the information, how viscous or how elastic your material, your sample is, that's going to define how your results would be. We all know that 2 hydrogels with the same stiffness might have a different viscous and elastic component. No surprise there. So what we can help you with, our 4 sensors or probes are provided from a wide variety of stiffness from 0.025, that's soft probes, up to stiffer as 300 nano per meter. Then the tips can be wide range of radius from 3 micrometer up to 25 micrometer. Is this enough, or what does it do? Let me make it simple for you. We can basically check the stiffness from 10 Pascal up to 1 gigapascal. That is a wide range of soft biomaterials covering from 1 single cell up to cartilage-like samples, with a wide range of force resolution, indentation depth, and contact radius. Now, how can we do this? What is this Optics11 Life Technology? Let's start with our nano-indenters, Puma and Tiaro. Puma, a benchtop design, literally inspired by a coffee machine. It is perfect for any sample that is like, let's say, up to 50 micrometer of dimension, hydrogel, tissues, organoids, scaffolds, 3D printed scaffolds. Just with a large working distance, you can just put the sample there, indent, measure, and go up to your next experiment. Not only that, if you are more keen into the single cell measurements, as Remi has been using, is the Tiaro. This Tiaro head can be combined with any of the in-house microscopes or even the special fancy fluorescence confocal microscopes. It is compatible and easily moved from one microscope to another as well. And this would let you, given the case, that you can measure up to 1 single cell, as small as that. Not only this, the recent addition, and today my focus would be more on Pavone. Pavone, let's figure out what it is. So no surprises there. It is a high throughput mechanical screening machine. It has high force precision with the possibility to combine 2 different well plates or 2 same. We even have temperature control and so much more. But what it can do, why Pavone? So the Pavone is designed really to tap into the science when we are going fast, right? Science is changing every day. Times are changing every day. What we need today is not one single measurement. We have cells throughout the well plates. Let's say 96 well plate, and you want to run a full-fledged experiment. You have the possibility to design a full experiment moving from one point, one well to another, and so much more using automatic fine surface, even running matrix scan at each point, doing dynamic testing as well. Basically, you can run overnight experiments. We have a touch interface with this, but the possibilities from indenting to imaging falls like this. For example, on the top left corner, you can see a mapping. What is this map? This is a map taken over hydrogel, a commercialized hydrogel that basically swells over the time. we started measuring a matrix scan using the autofine surface feature, we could see that we actually visualize the Young's modulus changing over the swelling period. Not only this, we could actually see how the topography was changing. Given that, if you look about the hydrogels and you want to do a matrix scan, organize tissues, all these measurements are possible. At the bottom left corner, you can even see a scan from a cell measurement. You can see this red part there, that is the stiffness coming from the nuclei, and the yellow region is actually from the stiffness of the cytoplasmic area of the cell. Similarly, you can even get the topography of the same samples. We can measure single cells, hydrogels, the limitation has no bound. Using the coordinate feature, what we can do, we can just using the touch interface, we can click on multiple cells. The machine would save the cell coordinates or any random coordinates of a hydrogel, and you can run a full-fledged experiment designed as per your requirement. Is this it? No. Let's say you want to do a quality control, right? You have a drug screening, you have a quality control for a whole batch of hydrogels. This is one of the commercialized hydrogel that we bought. It's from Matrigen software, going from a gradient of 0.2 up to 50 kilopascal. And the full 96 well plate was measured using one single probe. Even the numbers shown among the replicates are quite close. You can see that the standard deviation is pretty good. Reproducibility is the key here. And in science, that is one of the reasons that you can show that how qualified your data is, right? Now, what is this Pavone that I have been talking about, right? Let me give you a picture here. This is Pavone in-house with a touch interface. If I raise the lid, you can see a combination of 6 and 96 well plate. I want to move to a 2 well. I select it, and stages will move to the respective well. Basically, you can design a full experiment going from one well to another. Not only this, you can either do matrix scans, single indentations, multiple indentations with a fully designed, whether you want to pause, move, or if you want to just do some specific wells as well. Now, one of the examples I would like to show, because I mentioned earlier about the drug screening, the potential of Pavone has no bound. This is the perfect advancement given the challenges we are facing in the mechanical field, whether it's mechanotransduction, whether it's biomechanobiology, and so much more, right? In this example, this is from the astrocytes, which are basically measured with no treatment to treated with cytochelicin. And you can see how the impact of cytochelicin was clearly significant over the no change from fasodil. This is one of the similar experiments, I think, with Remi was showing that how the treated and with piezo and non-treated were showing. In this scale, the difference is only that the measurements are done on a very high scale. That means high throughput screening. This can be implemented like I said, for hydrogels quality or a drug screening or testing your cells among different, let's say, different controls, different treatments, and so much more. Is Pavone the only solution for your research? Probably not. One of our users has Puma, and they published this work, and they are actually measuring alginate gelatin hydrogels for cartilage repair. They are trying to understand the mechanical properties of the sample of the period of the 14 days. They see how the sample has changed during the first 24 hours and in the coming days to understand whether you can say it as how the sample is degrading or how stable it is for the reason that this scaffold has been tested, right? This could also be interpreted as, imagine you want to do a drug release study and you want that encapsulation to degrade over the time, right? So you would want your capsule to degrade within a span of maybe 1 week so that the drug is released continuously. Similar experiments can be done based on what your application is. Not only this, another user, as well with Puma here, our baby coffee machine, they are assessing the nano-indentation in stiffness measurement of soft biomaterials, and they go from kidney, liver, spleen, uterus. Look at the variety of samples that have been tested with one machine. Not only this, another user has Tiaro. They are testing cardiac fibrosis in vitro. So this work is not only mentioning about cell stiffness, but it is also mentioning about the relaxation and adhesion forces, which I really like because they're really taking the information out of all the scales. So with that said, I'm curious what you might be acquiring next time you're looking for a nano-indenter. But my take-home message here would be, cells, tissues, hydrogels, anything can sense and respond to mechanical microenvironment. So it is the first property that you might look into when you're looking for anything else. Mechanics can also act as a biomarker from healthy to unhealthy, let's say, or degradation, and so much more. Above all, automation or the high throughput screening. So to be said, possibilities are endless and we have the solution. I really hope it was useful to you. And if you're really interested, we are happy to have you as a consultant call. We are happy to test your samples and see which equipment fits you best, and so much more. Now, with that said, I would like to ask you a few questions. As let me launch a poll, and I want to see what you're really interested in. Let me start with first, that is, what samples do you work with? Hydrogels, cells, hydrogel spheres, microspheres, tissues, spheroids, or organoids? I'm sure someone is working with something. Nice, I will give a minute more. I do have a shy audience today. My smile didn't help, I guess. Oh, there we go. Now we are opening up. Okay, a wide variety, I would say. Everyone is working with something. So you can see it for yourself. The results are there for you. And you can see that everyone is falling in similar category, basically. And indeed, those samples are measurable using our indenters. Now, I'm curious that if these are the samples, and if you have seen the presentation, which instrument you might like to acquire or you think is useful for you? Don't be shy. I saw a lot of answers. Okay, so I basically just have to say it every time. Okay, interesting. Anyone last minute? Oh, I see the polls changing. Puma, Chiaro, Pavone. Great. I got a very, very distributed answer. So Puma, Pavone, or not sure. You know what? I'm gonna share one more poll just because there is a not sure. But anyway, you can look it for yourself. I'm not lying there. Yeah. So given that, I have one last poll. I assure you, I'm gonna leave you after that. So what would be the next step? If you're not sure, would you like to test your samples? Or the people who are sure, would you like to receive a code, or you want to have a call with us? How would you like to proceed? My shy audience, come on, not shy anymore. Yes, okay, very clear. No, not anymore, 50-50. Anyone last minute? Great, okay. Interesting. I would share the results. So basically, people want to test their samples. So we can definitely arrange for a demo with you guys. I would be happy to help you out. You can whether contact us at Optics11Life website, or you can just send me an email at priya@optics11life.com. For the materials also, we would be happy to share. And if you're really not sure, the one last thing I would like to share here is that we can always discuss, because it's not about just some equipment. I think why we are discussing or why we want to talk to you is to understand what you really need, because that is the question that we need to answer. If you are unsure, or if you are thinking, like if you have any doubts, we need to understand you are running away from next level of research. Is it just because you have seen complications, or you expect that it's going to be tough, or there are limitations, obstacles? If that is the reason, then maybe that's not the reason anymore. And apart from that, given the application that you might be working in, just a call can probably help you. Without even the equipment, you might take it to the next step. And if it's the equipment, we would help you figure out which one is the right fit for your application and the goal. I hope it was useful. And once again, my special thanks to Remi for joining us today. Thank you so much, Remi.
So I saw that no one is interested in the Tiaro, and I wanted to add something. So I work a little bit with IFMs and so on, but one of the most interesting points from what Optics11 can bring with the Tiaro system, it's very easy to use, and it's extremely flexible. So we are interested in electrophysiology. We are interested in getting plenty of different readouts. So we want to stick in probes. So patch pipettes, stimulators, perfusion systems. We want to work with different angles and so on, with different materials. And this is what IFMs are really not great at doing, as far as I know. So being able to work around the nano-indenter is a fantastic opportunity, I think. You can stick whatever you want, wherever you want. And if you're not happy with the orientation of the cantilever, you take your screwdriver and you change it. And I really, really like this flexibility.
Well, we have someone explaining why they did not select the Tiaro. No worries. You can still get a Tiaro. No worries. I cannot launch the poll again, but I hear you. But thank you so much, Remi, because this is what I think I personally love. Being a scientist myself, this is what I love hearing from the users. Because as a company, we can say that. But given that you have first-hand experience for so many years now, and you have known the company for a quite decent long time, and you are using Tiaro so regularly, there is nothing more that speaks than your own experience. So I'm personally very happy that you actually feel like that. And yeah, if I had to buy one, I would actually buy Tiaro. Probably because I love microscopy as well. But thank you so much, everyone. Please don't hesitate to reach out to us via email, via our info@optics11life.com forms as well. We would be happy to help you and find that missing piece. Thank you so much, everyone. Thank you, Remi.
You're welcome.